Happy Saturday, scientists. Let's review. Although you and I are both humans, we're different. In today's topic, we're going to be learning about how these differences come about, how humans have influenced the development of these differences in other species in the past, and how we're influencing them now. Today's topic is B5, Genes, Inheritance, and Selection. Let's begin. We begin once again with a great selection of keywords to start us off. Variation is the difference between the different individuals of a species, and this can be genetic variation, so what's coded for in your genes and what you inherit, or environmental variation, which is based on where you live. Some things are a combination of both, such as your height. If you don't have the right nutrients, then even if you have genes to be tall, you won't be able to reach your full potential. There are a few more definitions, however, I'm afraid. Phenotype is the characteristic that an organism shows. So if you are looking at an organism, the phenotype of that organism is what you're going to see. And this is different to the genotype, which is the genetic makeup of an organism. In terms of variation, we have two different types. We have discontinuous variation, which is when the variations fit into distinct categories. Such as eye color or hair color. But we also have continuous variation, and this is when variations are able to be any different value. An allele is one copy of the genetic code, or one copy of a gene. And you will have two alleles, one from your mother and one from your father. A dominant allele is an allele that shows its phenotype over the others. Whereas a recessive allele is an allele that only shows its phenotype when there are two of them. Heterozygous and homozygous are two words that discuss which alleles you have. The zygous termination means that we're talking about the zygote, or what's inside the nucleus of the cells, and heterozygous means that you have two different alleles. You'd have one dominant and one recessive. Whereas homozygous means you have two of the same allele. The next thing we're going to talk about is meiosis, which is the process of producing our gametes, or our sex cells. So in the case of humans here, we're discussing the sperm and the egg. Now, there's a few more keywords to learn here, and the first is diploid or diploid. And what that means is cells that have two full copies of the genome. Versus haploid, which are cells that only have one copy. In your body, you have mostly diploid cells. You've got one copy of the genes from your mother and one copy of the genes from your father. However, Your sex cells have to be haploid because their purpose is to join with another haploid cell during sexual reproduction to form a diploid individual. And so for that reason, meiosis is the process of taking diploid cells and turning them into haploid cells. In humans, this means we're going down from 46 chromosomes to 23. Now the 46 are made up of 23 pairs, which means what we're going to do is we're going to be splitting those into their individuals. So one part of the pair of chromosomes will go to one cell, and one part of the pair of chromosomes will go to another. I find that it's useful to know the names of these stages and how they fit together to form kind of the cycle of cell division and cell life. You may remember that we have a phase called the interphase, which is sort of a rest phase, where the DNA is kind of in the nucleus and is kind of spread apart for its actual use in the cell. What we're then going to do is we're going to copy the DNA. And what that will mean is that when we enter the prophase, we are going to have a set of DNA that is complete for both cells. And what they're going to do in the prophase is the chromosomes are going to pair up. So if we have a cell with simply a couple sets of chromosomes, then in prophase you'd have the originals and the copies paired up just like this in the nucleus of the cell. After prophase, what's going to happen is that the chromosomes are going to line up in the center of the cell. And this will put us into metaphase. Now this should be fairly familiar to you because it is the same steps as mitosis. However, because we're trying to go from a diploid cell to a haploid cell, we are going to end up going through this twice. And so we label these each with a 1. So it's prophase 1 that we've just been through, and we're now in metaphase 1 with our cells' chromosomes lined up along the center of the cell. And they are ready to be kind of pulled apart to the far sides of the cell, and that's going to happen during the anaphase. We're now going to have our chromosomes being pulled apart to separate sides of the cell. And this is anaphase 1, chromosomes pulled apart. From there, we are going to start the division of the cell into 2 cells. So we're going to end up over here in what is known as the telophase. And again, it's telophase 1. And here, the cell is going to start to pinch in the middle in order to form new cells. And you're then going to have your new cells with their DNA in their nucleus, which starts to reform at this point. From here, we are going to go into prophase 2, where we'll have the DNA lining up again, or coiling up rather, so forming up into its chromosomes. In each of these new cells that we've formed, you've got your DNA forming in the nucleus into its chromosomes. You've got the fact that the nuclear membrane of these cells is going to start to disappear as well as we get ready for metaphase 2. I think we can guess what's going to happen here. You're going to have the DNA lined up along the middle of the cell. From here, we're going to enter anaphase 2. where you're going to have your DNA pulled apart to separate sides of the cell. And this is really the point at which we become haploid cells. So we've now kind of separated out this DNA. And then finally, we have telophase 2, wherein you're going to have your cells dividing to form 4 daughter cells each, which is genetically distinct. And so here we'll have the formation of the daughter cells with their chromosomes in them, and they will be genetically distinct to each other. And this will give us the 4 haploid cells at the end of meiosis. What this means then is that we will be able to recombine these haploid cells through sexual reproduction in order to achieve a crossing of genetic material. And what we will be interested in then is what are the chances that a particular characteristic happens. Let's use an example of cystic fibrosis, where you have true heterozygous parents for the alleles, and it's the recessive allele that causes the disease. We show the recessive allele with a small letter and the dominant allele with a large letter. You would do either a Punnett square like this, or you may do a genetic map, and what you'll do is you'll see what the cross actually is. So here we'd receive these 2 alleles if this is the random chance that happens. In this one, we'd receive these alleles. In this one, these alleles. And finally, in this one, recessive alleles. So this means that there is a 1 in 4 or a 25% chance of the disease phenotype. This doesn't guarantee that if a couple had 4 children, there would absolutely be one of those children that has the disease. They could have all 4 healthy or all 4 with the disease. It just means that there's a 25% chance of that being the genetic combination between the 2 cells. Now, all of these genetic combinations are caused by mutations, which are changes in the order of the base pair. Some of these are neutral, so they don't change the protein that is made. Some of these are positive, and so they change the protein in a way that helps the individual survive, or that helps the individual reproduce. And some of these are negative changes, so these are changes that harm the individual. Mutations will happen naturally, or they will be caused by mutagens in the environment. The word mutagen just means mutation generator, and so this could be things like UV rays, certain chemicals, etc. And these will cause mutations and therefore could cause harm or sometimes won't change anything in the individual. What these mutations lead to is the principle of natural selection. And this means that gradually some individuals, those that are best able to survive and reproduce, will be the ones who pass on their genetic material. What we might see here is, for example, in a population of bacteria, you may have a couple bacterium that are actually resistant to an antibiotic. As time goes on, you'll have less of the bacteria that aren't resistant because they'll be killed off by the antibiotic, and you'll have more of the bacteria that are resistant. Eventually, you may lead to evolution, wherein a new species is evolved because those are the only individuals that are left, or the only individuals that are left in that region. If we're talking about individuals that use sexual reproduction, then a new species is when those individuals are so different that they can't reproduce to produce fertile offspring with the others. And so that would mean that you've evolved a new species because they were best adapted to the environment that they were in at that point in time. And there's a number of different ways that we've been able to find evidence for this. We've been able to find fossils, which are when minerals replace the tissues in an organism, or replace some of the compounds in the organism. If, for example, you have an animal that died, here is my attempt at drawing a skeleton. You'll notice that it is extremely talented. So there's our skeleton, that animal died, and eventually the bones here will be replaced with different minerals. And what will happen is that each year there will be new material added on top of our skeleton. So there'll be a new layer of soil. And eventually, the skeleton that we have is going to be buried under many, many layers of soil. And we'll be able to dig down and to essentially go back in time. If we know that there's now a different animal living in that area, maybe it's a taller animal, we might be able to go back in time and actually find fossils where that animal was shorter. And maybe in between we find an animal that's sort of in between them. We can see that this original individual, this original species, evolved into today's modern species because having a taller individual was selected for, or was an advantage for natural selection. The species developed over time. You can also see this using molecular evidence, so using the DNA sequences of individuals. So you can trace back to when a particular mutation happened, and then you're able to see how that mutation has happened in a bunch of different species. And so you might look at it and say, well, maybe that mutation happened at the same time in all these different species, but it's more likely that they came from a common ancestor. And so that's where you'll start to see these trees that you might have seen developed, where you've got individuals branching off from common ancestors, and that's going back in time as you go back down the tree. What this means is this individual here is a common ancestor. And that means that in the population of this individual, some of them had mutations that took them this way, and some of them had mutations that took them that way, and eventually they formed different species. But those species are related by a common ancestor. There's something that they have that they might share in common. So the common ancestor might have had the mutation, and it went one way in one group of individuals and another way in another group of individuals, based on how they were best able to survive. All of this leads to a little bit of problems when we go to try and name something or to try and organize something. And so there was a scientist named Linnaeus who developed a way of classifying and putting all these organisms together. The system developed by Linnaeus was a way of categorizing individuals or different species and then of naming them. What he did was he started with large categories and got more and more specific as he continued on. So let's use the example of the house cat. And its Linnaean name, or its binomial name, is Felis catus. If we think about the largest category that this could possibly belong to, that would be what we would call the kingdom. If we think about what kingdom the cat belongs to, well, it belongs to animals. And next we want to think about what kind of subcategory of animal this is, and cats are animals that have a spinal cord. So they're divided into the next group, which is called a phylum. And so the cat is a chordate, meaning that it actually has a spinal cord. And now we look at all the animals that have spinal cords, and we need a new subdivision. So the new subdivision is going to be the class. And here it's going to be a mammal, because cats fall into the category of mammals. And now within mammals, we need to have another group. So we're going to have a group of animals that eat meat. And so this is going to be the order, and these are going to be the carnivores. Underneath that, we are going to need to have a family. And these are a group of much more related individuals. And so here, the family for cats is going to be the Felidae. And finally, we come to the last two, the genus, which in this case is Felis, and the species, which is Catus. And so these final two are the binomial name, but with them they carry the meaning of the rest of it, of the rest of this order. So starting with the largest division, the kingdom, and going all the way down to the specific, to the species, that lets us know where the animal falls in this category. And so now that you know about the variation that exists within species and that it is caused by mutations, which can either occur naturally or be caused by something in the environment, this is a favorite topic for 6-mark questions on exams. So how did X species evolve from Y species? And so you need to talk about those adaptations and natural selection and things such as that. There is a revision worksheet, as always, linked in the description of this video, and the answers to that will be out next week. If you have any questions, please leave them in the comments below. Until next time, happy sciencing.